This video is going to be about how local conditions affect enzyme activity. So we know that enzymes are proteins, and we also know that proteins are pretty sensitive to their environments. So there's going to be several different factors which can affect the activity of an enzyme. So two of those factors are pretty important in all sorts of protein biology, and that's going to be temperature and pH. So we know from the video on protein denaturation and protein folding that when the temperature gets too high, the protein can actually lose its shape. So with enzymes, it's a little different in that increasing the temperature typically will increase the rate of the reaction with that enzyme up to a certain point because the molecules will be moving faster, so you're going to have the enzyme coming in contact with the substrate more frequently. But once you get to a certain temperature, that's not going to be the case anymore, because then the hydrogen bonds within the protein are going to start breaking down, and so the protein will actually become denatured and the enzymatic activity will be absent. And so moving on to pH, just like temperature, enzymes have an optimal pH where they work the best. And for most human enzymes, that's going to be around a pH between 6 and 8. But there are some exceptions, like enzymes that we would find in our gastric juices in our stomach. So those would have to be active at a much lower pH than other enzymes in our bodies. So moving on to something called cofactors. So cofactors are basically just a molecule that kind of helps the enzyme perform its function better. So these cofactors can be bound tightly and permanently to the enzyme, or they can be bound loosely and reversibly and come off and on as needed. So cofactors are typically going to be inorganic molecules, but if you have an organic molecule, which is going to be something that has carbon in it, so if you have an organic molecule that's acting as a cofactor, then that's actually called a coenzyme. So just to review, temperature is typically going to increase the rate of a chemical reaction until the temperature gets so high that the protein becomes denatured. pH, just like temperature, there's an optimum pH range for human enzymes, and that is typically between a pH of 6 and a pH of 8 with some exceptions. And then inorganic molecules that help an enzyme perform its function are going to be called cofactors, and then the organic versions of cofactors are called coenzymes. I hope you found this video really helpful. The concepts and information presented in these videos will be true regardless of what biology course you're taking. However, the material we covered in this video is specifically referencing material covered in Campbell Biology's 11th edition. Remember that if you are an enrolled Baylor student, we do offer free tutoring on the first floor of the Sid Richardson building, and you can schedule a free 30-minute appointment to have one-on-one tutoring online, or you can stop by during any of our business hours. For more information about the services we provide, you can go to our website at www.baylor.edu/tutoring. Thank you.